Hi, my name is Robert Sapolsky. I'm a professor of neuroscience at Stanford University, and you have just bought yourself a user's manual for your brain. What we're going to be doing over the next 8 lectures is looking at how your brain works, how your brain influences events throughout your body, and what this has to do with your individuality. Okay, what do I mean by this? Let me start off with some scenarios that will hopefully convince you this is something you got to learn about. First unlikely scenario. 40-year-old guy living a quiet, peaceful life in his suburban home, working in the office each day. 20 years he's been married happily, kids running around in high school, 2 dogs, 2 cars, that sort of thing. One day, suddenly out of nowhere at the office, he gets into a fist fight with somebody. Fist fight with somebody over some minor deal. He hasn't had a fight since high school. Inconceivable. He punches someone in the nose. 6 months after that, his wife of this 20-year marriage discovers he's having an affair with the 18-year-old checkout kid down at the supermarket. Even more strange things happen. 6 months go by and he's arrested for brawling drunkenly in a bar. He never even used to drink. This is what he's doing 4 nights of the week now. Another 6 months go by and he embezzles all the funds from his business, disappears, is never heard from again. Okay, what do you make of this? What we mostly think looking at this is one of the more dramatic midlife crises one could see. This is one of the more exaggerated versions of somebody really becoming childish and destructive. And what if instead I suggested to you that this might be due to a genetic disorder? That there is a disease out there where there is a single amino acid in one protein somewhere in the brain of this individual, and that genetic defect has been there his entire life, and it's programmed so that approximately 40 years into life, there's this dramatic behavioral change. Is this somebody just really going to hell very dramatically and falling apart, or is this a genetic disorder? A lot of evidence to suggest it could be the latter. Okay, another scenario that's just as disturbing in some ways. Here's something I know we all do. We have days where you're a little bit anxious about something and you find yourself counting the number of stairs when you go up a flight of stairs. You find yourself, there's some stupid TV jingle in your head and it's stuck there all day. You cannot make it stop no matter what you do. Or I know we all have done this one at some point or other. You've got some important letter to send off, some application, something or other. You go over to the mailbox and you put the letter in there and you make sure it's gone down, and because it's important, you check again and you're about to go away, and maybe you check the mailbox again and you look around and there's nobody else around, so just to make sure you look at the bottom and you look at the front of it and you check 47 times to make sure that letter has gone down. Yeah, we all have these quirks like this. We all have these days where we get stuck in these little behaviors. There's a whole other neurological disease out there where people are trapped in these patterns forever, where people spend 6-7 hours a day washing their hands, where they can't go into a room without turning the light on and off a even number of times between 12 and 17, a multiple of the day's stock market quotes, where they're just caught in these little rituals. Another neurological disease where people are beginning to understand there's a genetic disorder in these folks, there's a neurochemical disorder that makes you check the mailbox 28 times instead of the rest of us doing it 3 or 4 times. This appears to be the case, though. Another example. You've got somebody who's just been diagnosed with a fairly serious disease, and this is a disease that's going to affect them for the rest of their lives. Not surprisingly, this brings about a couple of behavioral changes in them. They suddenly find themselves becoming a lot more serious about life, taking an interest in some philosophical issues. They get a little bit less of a sense of humor than they used to. Maybe they get a little bit more introverted about life with their new illness. They're not as willing to try new things. They fall back on familiar patterns. They're more dependent on old friends. Maybe even they become religious now when they didn't used to be. Okay, that's not surprising. Serious illnesses change the way we behave. What if I told you there was a neurological disease out there where it's the disease itself which causes these behavioral changes in people? A certain type of epilepsy where people with this disorder, independent of how well controlled their seizures are, independent if it's been years since they've had a seizure, they get a personality change with this type of epilepsy. They become humorless, they tend to fixate on things, they prefer familiar old things, they don't like new things, it's called a neophobia, they get very dependent on old friends, And this is the most disturbing thing of all. A consistent feature of this personality disorder with this type of epilepsy is people become religious at that point. And this is no more severe of a neurological disease than many others. This is the disease causing a change in who that person is. This is very bizarre stuff. We are very accustomed to thinking of ourselves as individuals, and we take a certain amount of pride often in this individuality, and there's a whole science out there suggesting a peculiar gene, a peculiar level of certain nutrients in the bloodstream, peculiar levels of certain hormones, peculiar levels of certain brain chemicals, and you're a different person. And that begins to challenge our sense of what makes us us. And this is important stuff. This is important stuff because all of us have to make expert opinions on these sorts of things all the time. You're sitting on a jury, and someone is there in the defendant's box, and this person has done something appalling. Is this because the person is no damn good, and you can actually begin to invoke concepts like evil? Or is there something wrong with a part of his cortex which makes him unable to control impulsive behaviors? You've got to decide if that person is going to be treated as someone who's criminal or someone who has a disease. Or another case in which we have an impact in this sort of way, you're sitting there and your spouse has been sunken in a depression for the last 6 months. Is this a neurochemical disorder that they can't control, or is it one of, oh, come on, you know, we all have our depressed times, get it together, stop indulging yourself? Is this somebody who's just allowing themselves to slide into a funk, or is this as real of a disease as diabetes? And none of us sit around and say, oh, come on, what's this insulin stuff? Stop babying yourself. Is depression as real of a disease? That certainly makes a difference how we deal with our loved ones when they fall into them. And we are all expert opinions. We are all experts and better have informed opinions when it comes to how we judge our children and how they're learning, and a child is not learning very well. Are we allowed to say words like unmotivated or perhaps even ungifted? Or is there a neurological problem? Is there a learning disability? Things of this sort. These issues affect every single one of us. We often lament these days about how we make these opinions built around what we should do with technology in our society when most of us don't even know how to fix a dry cell battery circuit. We're deciding should there be nuclear power, things of that sort, when we're woefully uninformed in that area. Even more so, every single day we are making decisions based on what is the biological imperative, what is the core of who we are, and what's the biology that impinges on it, and we'd better understand how that stuff works. Because there's an amazing science out there that's impacting what makes us us. And the really striking thing about that stuff is, at some point, all of that information that's being accumulated, it stops being a science of them and their diseases, and it gets real close to home. It's a science of us and what makes us us. And this tends to get people real uncomfortable at that point because it gets close to home. The core of this, the centerpiece of this whole business, is the fact that there's this intertwining between the chemistry, the structure of our brain, and how we behave, how we feel, the ability of events in our body to influence our mind, the ability of our mind to influence events in our body. All of this very critical. Couple of obvious examples here. First one, in terms of how our mind can influence events every spot in our body. Here's an exercise. Okay, we've all done this one. Make this very clear this time. Tonight, when you're lying in bed and you're nice and relaxed and you're just about asleep and, you know, you're listening to your heart beating nice and slowly there, try the following thought. You know, that heart isn't going to beat forever. And I bet you at that point your heart rate's going to increase. And you won't have just run up a flight of stairs, and you won't have just been escaping some lion chasing after you, but your physiology is different at that point. Think a thought, and you're changing events in your big toe, and your spleen is faxing some hormone you've never heard of to your pancreas, and thought affects your body. Conversely, what's going on in your body affects your thoughts. And here's a very interesting example. Okay, think about this. I will give you 4 circumstances, and they all have something very important in common, very relevant to this course. Number 1, having a brain tumor. Number 2, having your period. Number 3, eating a lot of junk food. 4, taking anabolic steroids. Okay, anabolic steroids, those are the steroids that weightlifters take. 4 circumstances, and they all have something very important in common. Having a brain tumor, having your period, eating a lot of junk food, or taking anabolic steroids. And the wild thing they have in common, which absolutely underlines the importance of the subject, is People have all been defended in courts of law throughout the Western world against charges of murder on the basis of, sorry, they've got a brain tumor. It's not them, it's their disease. Sorry, it's a well-known fact that people become more aggressive during their perimenstrual period, right around their period, premenstrual syndrome. Sorry, your levels of glucose in your bloodstream can influence your behavior, and there's a subset of us for whom eating junk food makes you more violent. Sorry, testosterone, male sex hormones, anabolic steroids can make you uncontrollably violent, and in a few of these cases, juries have decided, yes, indeed, this is not someone who is evil, this is not someone who is violent, this is not someone who is anything other than being distorted by a biology going on there. Totally independent of whether you think that's a good idea or not, what you need to know is how this stuff works, because this affects all of us. And that's what we'll be focusing on in this course. What is the biology of how our brain works, how it influences every far-flung cell in our body, most of all, what this has to do with behavior. And our theme in lecture after lecture here is going to be working through one of these physiological systems, working on the links between the brain and our body, focusing on what it has to do with behavior, and most of all, trying to understand why this may begin to teach us why you and I are different, what the biological bases are of our individuality. Okay, the general strategy is going to be we're working from the bottom up. We're working starting off at the beginning at the small level, how does one single brain cell work? And that's going to be the focus of today's lecture. Slowly building out from there. The next lecture will be how do two brain cells, two neurons, how do they communicate with each other? And that has a lot to do with brain chemistry, chemical messengers that are released by one brain cell that go to another one. That'll be the focus of the second. Building outward from there, we're going to see how the functioning between these pairs of neurons can change over time. That sounds rather abstract. That has a very important real-life analog. Changing function between two neurons over time is what we do when we learn. And some of us do that better than others, and that's mighty interesting stuff that's going on there. Moving outward, we next go from the level of two neurons and how they interact to whole networks of neurons and how you wire them up to start doing clever things for you, how 50 neurons can begin to detect visual patterns, how they can do different types of sensory information, how they might do even more subtle types of memory formation for you. Really interesting stuff there, and very important implications for diseases where our memory fails, like Alzheimer's disease, things of that sort. That will get us ready for our next jump, which is now going from the level of 50 neurons at a time to millions of them, and a whole section of our brain that specializes in telling the rest of your body what to do. And that's the business where you think a thought and your heart speeds up, you think another thought, you flush, you break out into a sweat, you have some sort of uncontrolled response, some involuntary response in your body, looking at what's called the involuntary nervous system. Boring part of your nervous system, this motor system, you shake hands with somebody, you sign a check, that sort of stuff. This involuntary, the part of your nervous system you normally don't have a whole lot of control over, that's the part that regulates cells throughout your body, and that's got a lot to do with why some of us are more prone to heart disease. Getting stuck in the same slow bank line. There's a subset of us who succumb to hypertension, a subset of us in stressful periods who collapse into ulcers, and that's got a lot to do with the workings of this part of the brain. From there, we will shift to the other way in which your brain can influence events throughout your body, hormones. Your brain turns out to be one big hormone-releasing gland, and this is something that wasn't realized until relatively recently, and your brain runs the hormonal events throughout the rest of your body, and that's another way in which think a thought, have a memory, have an emotion, and you can reach out and influence the activities of every cell out there. Okay. That will give us basically the building blocks from the level of a single cell to whole regions of the brain regulating events throughout the body every single step of the way at each one of these levels of explanation. The goal by the end of that lecture is going to be where we could at least begin to guess a little bit how this works differently in some of us, how this may underline certain behavioral diseases, even more so, how this may underline individual quirks and individual idiosyncrasies, the biology of what makes us us. And in the final lecture, we will basically build on all of that knowledge from the single cell level up to the systems level to look again at some of these behavioral disorders, these diseases that I alluded to just now at the beginning, and a bunch of other ones, and these are absolutely bizarre ones, and these are ones where normally people sit there and say, nah, that couldn't be genetic, that couldn't be biochemical, that couldn't be due to something going, that's who the person is. By now we will have a very good sense of what the biology of some of these disorders might be. And following that, I think when this is all done with, it may not be that hard for a lot of you to sit there and somewhat objectively analyze your own little quirks and begin to get a sense there's a biological roots of everything ticking away there. We are the sum of all those neuronal connections and chemical messengers and things of those sorts. And the final thing we need to do is deal with the ways in which we get disturbed by this, because this challenges a lot of folks' philosophical senses of who we are. This challenges a lot of folks' sense of how we make decisions and judgments in our society, of who we educate, who we medicate, who we jail, things of that sort. These are important subjects. One thing I need to emphasize right from the start, amid all of this being state-of-the-art modern neuroscience, is most of you out there are probably not state-of-the-art modern neuroscientists and don't particularly wish to be in the next 8 segments of 45-minute tapes. This is not going to be painful. The marvelous thing about this stuff is this is accessible to anyone, and this should be accessible to anyone. I can almost guarantee you the scientific content of this will be easy and accessible. And I can almost guarantee you the implications of this are not going to be. If you're not disturbed by some of this stuff, you're probably not listening very closely. This is really disturbing stuff because it really challenges some of our most dearly held beliefs about us being these wonderful, mysterious individuals. There's a lot of mechanistic stuff going on underneath there. Okay. So we're ready to start now. We're ready to start with the actual nuts and bolts. And today being what the brain is built of, the individual cells. And starting with our first diagram here, we have the basic constituent of your nervous system, a brain cell. A brain cell, as most of us know, the main type being called a neuron. The neuron is your basic building block of the nervous system. It's not the only type of cell in there, and we will see some other types shortly, but this is the meat and potatoes of your nervous system. Your neurons, your brain cells, we've got a mere 100 billion or so in our heads, you know, plus or minus 50 billion or so, lots of debates on the exact number. There's a lot of them in there. And this is your standard neuron. This is a highly schematic one, and of course no neuron on Earth actually looks this way, but this is your basic schematic one and certain parts. And I guarantee you this is all the jargon you're going to get for this lecture, but you will come to know these terms and cherish them. These are going to be the building blocks of understanding how one cell functions at a time. All right, as is required by law, all neurons go from left to right, and that's the flow of information. Show me a textbook in which neurons go from right to left and something's desperately wrong there. All of it goes this way. That's the standard form. Information jumps from one neuron to the next. And we'll see in the lecture how, in the next lecture, how this occurs. For the moment here, information is moving within this cell, within this neuron, from the left end to the right. This neuron has just gotten some wonderful news. It's just heard some great gossip from the neuron to the left, and we will see how the neurons gossip with each other. This neuron is excited about this now. Quite literally, what that excitation is about is electrical excitation. And I guarantee if you hated physics in high school, that's the last you're going to hear about electrical excitation. It's not going to be more detailed than that. This neuron, nevertheless, is electrically excited. There's a wave of excitation sweeping from the left end to the right end. Of course, it's not that simple. Where the information comes in is through these little fibrils, these little tenderest little nets of information, of little cilia sticking out there, these things called dendrites. Dendrites are the ears of a neuron. That's where the neuron is listening to the gossip coming in from the preceding brain cell. That's where, as we will see in the next lecture, the chemical messengers released by the previous neuron float across the small gap and influence events in this neuron. The dendrites are the ears. As a result of the dendrites hearing this exciting news, this gossip, this chemical messenger, this elicits this wave of excitation, this wave of, technical term, depolarization. All sorts of chemicals, ions, are flying back and forth across the cell membrane. Things that are normally kept outside are going inside. Things that are inside are going outside. This is where stuff is occurring. There's this wave of excitation sweeping down the cell membrane. That's great. inputs from various dendrites and sweeping down the cell membrane across the cell body. This is the main business end of this neuron. Centered in there is the nucleus containing DNA, and this is a schematic version of DNA that we will see popping over and over. This neuron is excited, that wave of electricity is sweeping over. It comes across this field of dendritic spines, these little tendrils, sweeping across there, comes to a critical point, a juncture, whose significance will become a lot more important in a little bit, an area called the axon hillock. Piling up more of this jargon, not too painful so far. We're up to the dendrite and the axon hillock. Axon hillock, something very different happens there, which we will see shortly. For the moment, though, the basic event is this wave of excitation continues to sweep down now this long cable called the axon. Axons are not to be trivialized. Axons are major league features of these cells. This is a remarkable feature about neurons. Neurons are gigantic cells from the perspective of your body. Neurons are huge. There are neurons sitting up in your motor cortex, a part of your brain, neurons that control the movement of your big toe. Some of those neurons will have axons that are 3 feet long. This is one single cell with an axon 3 feet long. I mean, cells are supposed to be things that fit at the periods at the end of the sentence. This is a cell with an axon stretching 3 feet. If you're a blue whale, you can have an axon that's 60 feet long in your spinal cord. These are gigantic cells, and this is the business end. This is where the long part is. The axon is this long projection. Interesting side note, one of the themes that will come up over and over again here is if you've got cells that are this big, cells that are huge are expensive. And cells that are expensive are vulnerable cells. That's why an awful lot of neurological diseases hit neurons in the spinal cord, diseases where you have trouble with movement, Parkinson's disease, Huntington's disease, ALS, Lou Gehrig's disease, motor neuron diseases. A bunch of these preferentially hit these long spinal neurons. If you got a long neuron, you got an expensive one. And those are ones that often fail. So this is where the cost accrues. You've got this long cable, this long axon, and that wave of excitation that we mentioned, that depolarization, is now sweeping down the axon. Comes to the very end. The very end, the far right, the end, the terminus of the neuron, not surprisingly, is called the axon terminals. And what you've got is a whole bunch of these little fibrous projections here, and this is where the neuron ends. This is where it talks to the next neuron in line. The axon terminals hook up to the dendrites of the next neuron on. And we'll see that hooking up is a rather fancy process. So we've got a first pass, a first very simple picture here. The neuron hears something exciting. Waves of electrical excitation sweep in through the dendrites, over the cell body, past the axon hillock, shooting down this long cable, down the axon, down to the axon terminal, where the message is passed on to the next neuron. Our first pass here. What you have also in red is a very useful trick. And this is one built around the other type of cell found in the nervous system, a cell that used to be considered utterly boring and structural and just backup stuff, something called glial cells. Glial cells used to be thought of as just kind of the superstructure, and they had the portotoilets in the nervous system, all sorts of temporary dwellings that glia set up when you were remodeling parts of the brain. Glia used to be the real backwater of the nervous system. They turn out to do all sorts of interesting things. Here's one of them. Glial cells can wrap around the axon, and the glial cells are shown in red here, and form something called a myelin sheath. And what a myelin sheath is, you've just insulated this wire. You've insulated this long cable. This axon is this big electrically conductive stretch of membrane, stretch of that cell. Glia form this insulation there. And what that does is make the whole thing work faster. Electrical waves shoot down axons a lot faster when they are myelinated, when they have a myelin sheath. And there's two ways in which you can see that proved to you in terms of being interesting. One is we're not born with these myelin sheaths on most of our neurons. We develop them over our early childhood, and a very striking thing is all sorts of behaviors suddenly start working in infants and kids right around the time when they myelinate the relevant part of the brain. Kids myelinate the part of their cortex where you understand language when you're about 10 months of age. And that's right around when kids are suddenly understanding stuff being said to them. 2 to 3 months later, you myelinate the part of your cortex where you generate language. Right on schedule. What's very good news for many of us out there, too late for me, but many of you still have a chance, we myelinate our cortex up to around age 30 or so. Parts of our cortex are not fully developed until around that age. You're only putting on myelin then, and suddenly pathways work a whole lot better. The other way in which you can see that this is a rather useful thing to have is to look at a disease state where you lose that myelin. And this is what you get in multiple sclerosis. Multiple sclerosis, for reasons nobody really understands, your immune system goes crazy and decides this part of your body is in fact something invasive that has as little business being there as a bacteria, blows away your myelin sheath, damages these glial cells, and these axons are now conducting those messages at 1/10 the speed as usual, and you've got problems at that point. A very useful part of the story. Okay, so we've got our first simplistic pass here. Electrical excitation sweeps in through the dendrites, over the cell body, down the axon hillock, and then speeding down the axon because of this myelin sheath, triggering the message, jumping over to the next neurons in line. Not surprisingly, it's all a lot more complicated than that. It's lots more complicated because of certain tricks that your nervous system has to do. One of them is built around, a very specific feature, very unique to your nervous system. All sorts of cells in your body communicate, and often in ways that are roughly similar to the way a neuron communicates with another one. Electrical waves of excitation. This is the theme throughout a lot of biology. Neurons do it in a way that's much fancier than anywhere else in the body. And there's a very simple reason for this. Okay, let me give you an example of why you need to have your nervous system be fancy. Here's your choices, two different scenarios. Think back to the day you hit puberty. Think back to that morning you woke up and that event that happened, and your parents had primed you with all those books that you had to read in advance and wanted to pay no attention to, and suddenly that day came and this was a big event, and your mother made you waffles and all of your relatives embarrassed you and that sort of thing, and this was a big maturational event. Tell me the truth now. Would it have made any sort of difference in your life whatsoever if you had reached puberty 24 hours later? Probably not. Second scenario. You step outside to get something out of the car, and unexpectedly, there's a tiger there that chases you up and down the driveway. And this is serious stuff. You were running for your life, and you increase your heart rate and your blood pressure, and your nervous system tells your body to get all sorts of flight and fright responses, and everything kicks into gear. Tell me now, how much trouble would you be in if your blood pressure increased 24 hours later instead of 2 seconds later? This is what you need a nervous system for. Puberty, growing, hormones, you could do it now, you could do it tonight, ovulate next week, it's not a big deal, grow antlers tomorrow, all that sort of stuff. There's not this pressure to be so scheduled in terms of time. You increase your blood pressure thanks to your heart, you do it at the wrong time and you're dead. You get your muscles thanks to your nervous system, your muscles uncoordinated and you're in big trouble. No part of your body is under pressure like your brain is to get things going fast and get things going coordinated. And we could see this schematically here. And here's the puberty scenario versus the running away from the tiger scenario. On top, this sort of event, something happens here, and much of the body works where you slowly have a response, and when it's over with, you slowly turn off. This is your world of hormones and enzymes being released in your gut. There's very little that goes on in your body where every second counts until you get to your brain. And the characteristic of brain cells and neurons is something happens and things turn on rapidly, and the minute it's done with, it turns off. For you bioengineering fans out there, the nervous system has a huge signal-to-noise ratio. It's real quiet when it's got nothing to say, and when something's happening, when those neurons have just heard something exciting and they've got to communicate it to the body, all hell breaks loose, and when it's over with, you turn off real fast. Neurons are expensive, neurons are fancy, because they've got this task like nowhere else in your body. And that's going to begin to explain some of the more complicated features of how these neurons work. One of these complicated features is shown schematically here. Looking at this whole issue of contrasts, when you've got nothing to say and you're the nervous system, you want to be real quiet, and when something exciting happens, you want to let a floodgate of exciting events occur. What that translates into is neurons have to really concentrate on contrasts. The difference between a neuron having nothing to say and a neuron when it's excited has to be as dramatic as possible. And what we see here in the middle is the schematic version of what would be unacceptable to a neuron. Here's a very idealized version. This is describing the sort of ions, sodium, chloride, terms you haven't thought about since chemistry in high school but don't have to anymore. These ions like that, how much of them are floating around outside the neuron, how much floating around inside. Here's the membrane of the neuron dividing the world between the outside and the inside, and this would be a state called equilibrium. The neuron's doing nothing in terms of work. Everything is just floating on either side equally. This is totally useless for a neuron. Neurons need contrasts. What they need to do is, when they've got nothing to say, they need to get certain ions completely on one side of the barrier, very dramatic separation, and when they've got something to say, when they are excited, they need to have an exact opposite state. Neurons have to spend a fortune generating these contrasts, because they need to be able to generate signals like this, an explosion of response and a very clear difference. You never want to have and be uncertain whether this is the time you want to increase your heart rate or not. Sure, there's a tiger, is this neuron excited or not? You want that neuron to be able to pull this off. And neurons spend a fortune energetically differentiating between their resting state and their excited state. The resting state is called the resting potential, and that's back to that chemical term, specifically referring to the fact that certain of these ions are kept very unevenly distributed on one side of the membrane. Certain of those ions are kept outside the neuron, certain of them are kept inside. For those of you who want to remember those horrible high school terms, sodium and chloride are kept outside, potassium is kept inside. The details aren't important. The main thing is the neuron is working like crazy to make this very distinctive state. There's no mistaking a neuron that's resting. And you guessed it, when that neuron is excited, exactly the opposite occurs. Channels open up, gateways, and sodium and chloride come rushing into the neuron, and potassium comes rushing out, and there's no mistaking the fact this is mighty different from this. This is a neuron that hasn't had a thought in its head all day, and this is a neuron that's screaming its head off because it's heard some important news. And that's what's going on at the dendritic end. The dendrites are the points where those channels, those sluices open up, and all sorts of these ions rush in and other ones rush out, and what you can detect is there's a sudden change in the electrical state of the neuron right around the point of that dendrite. Here we have a very special case here. We've got one single dendritic spine here that's just heard some exciting news, and here's that wave of excitation coming in. That wave of excitation, which is due to the fact that you've opened up those gates and these ions are moving all over the place. This is what things are like during the resting potential, and this is when excitation is occurring. There's no missing that fact. That neuron has just heard something important. At this stage now, one of the complications comes in, which is you've got this neuron that has this excitation way out at this end of the cell. This dendritic spine, this outpost way out there, has just heard some very important news. The very important thing at this stage is, it turns out that excitation is going to dissipate over time. That excitation is going to come ripping down that dendritic spine and start coursing in this direction and that direction. And as you go further and further from the spot where that gossip came in, that wave begins to decrement. It gets weaker and weaker. And what we see here is measuring, a thousandth of a second later at this point, that wave is decreased somewhat. A thousandth of a second later, it's even smaller, and it disappears at some point. What's this all about? Think of this metaphorically. You've got some pond, and you've just thrown some pebble in there, and you've got a ripple that's heading outward and getting smaller and smaller and decrementing. A very important implication of that. That wave is going to be long gone and forgotten well before it hits the axon hillock. A critical implication. No single dendritic spine, no single outpost in the neuron has the power to send a wave of excitation all the way down to the end. It dissipates over space and time. No single input to a neuron has the means to tell that neuron what it's going to say down the line. Instead, you have this weird sort of democracy going on in a neuron, a constant voting, a constant summation. You need inputs going on all over the place. You need lots of these dendritic spines getting excited all at once. That's the means by which you suddenly have enough inputs coming in there to get the neuron excited enough, to get that signal heading all the way down to the end. Any given dendritic input decrements over space and time. What you have to have instead is this property of summation, either what's called temporal summation or spatial summation. Either you have to have the same input triggered over and over and over and over again so you get enough of these waves that they finally sweep down the axon, or you've got to have enough different inputs, enough different dendritic spines being stimulated at once. One single dendritic spine stimulated once has next to no power in the system. You've got to have integration. You've got to have lots of different voices coming in there, all agreeing at once. A neuron cell body is an integrator of the inputs of all the different neurons around there. And this is real striking. You need to get enough of a wave of depolarization of this excitation reaching the axon hillock for something very exciting at that point to occur. All of the stuff I just described to you are the events going on from the dendritic spines up to the axon hillock. And the story up to that point is individual inputs decrement over space and time, so you've got to have agreement amongst a lot. The minute you get to the axon hillock, something very different happens. The axon hillock is sitting there and has this very different property. The axon hillock is the threshold. The axon hillock is the bean counter. That's the one that adds up how many votes are coming in, how many dendritic spines are getting excited all at once, how many of them are repeatedly being stimulated. It's sitting there counting on its toes, and the axon hillock has this rule: if and only if a wave of some particular size reaches me, then I get excited. The axon hillock is this point at which, if you get enough inputs, if you get enough excitation, if you get enough different dendritic voices in your town hall meeting all at once saying, yes, this is exciting news, suddenly your axon hillock gets excited and it gets a huge, explosive wave of excitation, far more dramatic than what was going on back in the dendrites. Enormous floods of these ions going in and out, all sorts of things. You have what is now called an action potential. And that's the most excitatory stuff a neuron has, and the property of the action potential is it doesn't decrement over space and time. It goes blasting down that axon, and that axon that could be 60 feet long, and it keeps regenerating an action potential of the same size, this huge wave of excitation saying, you can't believe the news we heard back there. All 999 of the 1,000 dendritic spines back in that end, they all say this is big stuff. Action potential goes sweeping down the axon, heads to the axon terminals, and passes on that exciting news to the next neuron. This is this very dualistic property of neurons from the dendritic end up to the axon hillock. You've got this decrement, you've got to integrate all these different inputs, pass that threshold, get the axon hillock, and you get this explosive wave, this action potential going down to the end, and an action potential that could be 5 miles long heading down an axon. It's going to be just as strong, just as regenerative is the term given for that, and you are going to have a very strong signal given out to the next folks. These are how neurons work, and obviously critical to functioning is this axon hillock. How jumpy is it? How much of an input, how much of an excitation does it take for the axon hillock to get excited? This is a critical feature, and different neurons at different times and different places in the nervous system have different thresholds. How much excitation does it require for that neuron to suddenly pass on that signal? Inputs from all these different ends, declining over space and time, ripples disappearing from the pond, hit that spot, and you've turned on a faucet. That's the action potential flying down. Okay, what is the most interesting implication of this axon hillock property, this integration that goes on here? In a lot of cases, this is not very interesting. A lot of parts of your nervous system you can do without this fancy integration of these different inputs going on here. What's shown on top, this is basically what your wiring is like in your spinal cord. And here we have a bunch of neurons talking to each other, nice, straight, linear system. One neuron sends all of its axon terminals to the dendritic spines of the next neuron, which in turn sends all of this. Moving on down the line, what you wind up getting is, if this neuron has an action potential, it stimulates all the dendritic spines on this neuron. That neuron's going to have an action potential, and that one's going to, and that one's going to, and you shoot down the line. This is boring stuff. This is your spinal cord, these very linear pathways. This is not the interesting stuff you do with your nervous system. Once you get into the brain, most of what's going on looks like this instead. Complicated networks where neurons are talking to each other. This kind of nervous system makes you move your finger. This kind of nervous system lets you write symphonies. And you're not going to be able to do that unless you've got these really complicated networks. Just to give you an absolutely horrifying sense of the numbers involved here, your average neuron sitting there in your brain has the ability to get axonal inputs, to have axon terminals terminating on its dendrites, axonal inputs from 10,000 other neurons, and to in turn send its own axon terminals to 10,000 other neurons out there. That's an amazing network and integration where every neuron is sitting there and hearing 9,999 other voices coming in there, no single one of which has a tremendous amount of influence, but each of which has a little bit of an impact. This integration, this sort of counting and sort of merging of inputs there. 5,000 of these dendritic spines are very excited right now, 4,000 are not. What do we think of that? Okay, what's my threshold as an axon hillock? 5,000 have to be excited at once. Yes, we've passed threshold. Action potential down the line, talk to the next 10,000 there. This very complex integrative network, this is how the nervous system works. And it better be wired up this way because we do fancy stuff. Okay, this gives us the basic building blocks of the nervous system. This is how one single neuron works. And what will be our task in all of these cases, and what we look at now for the first time, is how does the stuff work differently in different individuals? Just on the level of single neurons. Back to these properties we looked at here, where might there be individual differences? And people make a whole industry, some of the more obsessive of neuroscientists out there, sit around and what they do for a living is count the number of little dendritic spines on neurons. And that keeps them off the streets and that keeps them quite happy and active, and they turn out to find out some very interesting stuff. We all have different numbers of dendritic spines. We have different numbers of neurons themselves, numbers of dendritic spines, numbers of connections. That begins to be rather interesting. Not only that, we can change the number of dendritic spines we have in our nervous systems at different points in life. If you have your cortex, the part of your brain involved in abstract thinking, if your cortex gets a lot of stimulation, gets a lot of environmental enrichment, those neurons grow more dendritic spines. And that used to be something that was thought to be just a function of kids, infants, developing nervous systems. That turns out to be something you can do your entire life, well into your 90s, environmental stimulation, and these synapses, these connections, these dendritic spines, there's more of them to work with. More connections, more complexity. Obviously, another piece of the story is that axon hillock. And this is a real interesting one. Insofar as the axon hillock is this integrator, that's the one that counts the votes, that's the one that decides whether all of these inputs and all of this complexity at this end is going to mean anything whatsoever, what your axon hillock's threshold is very important. And it turns out that threshold can change over time. Hormones can make certain axon hillocks more jumpy or less jumpy. More jumpy, what do I mean by that? 10,000 dendritic inputs, and it used to be you need 8,000 of them to get excited at once. A more jumpy axon hillock would now be one that requires only 2,000 of them to be jumpy. That would be a more excitable neuron. One example in which hormones might be doing this, testosterone. Testosterone, male sex hormone, there's a certain part of the brain that's very sensitive to testosterone, and bathe that part, soak that part of the brain in this male sex hormone, and you get individuals who are more aggressive. In a subtle way, we will see more details about that later. It's not just that they're more violent, they're more readily provoked. On a cellular level, there's some evidence that what testosterone is doing is making axon hillocks in those neurons more jumpy, more excitable. And there's a whole science out there about how different of us differ as to how our axon hillocks respond to inputs, what the thresholds are, how readily hormones and neurochemicals and nutrients and things of that sort regulate this step. We begin to see a scenario that you can describe one individual with very different levels of dendritic spines, numbers of neurons, axon hillocks. Those are brains that are going to work differently. This is a first pass. These are the first simple ways in which we can begin to imagine this biology that makes us different. All you have to do is look at a cross-section, a slice of brain tissue under a microscope between two different individuals, and every single one of these variables differ, and they matter. Our first sense of what this might be about. What we need to do in the next lecture now is jump to the next step. We've managed to generate action potentials. We've managed to shoot information down this axon, down to the axon terminals. What we need to look at next is how the neuron is going to send on the signal. And this moves us into the world of chemical messengers between brain cells, neurotransmitters. And there's an amazing amount of information known there and ways in which we differ in our neurotransmitter levels. So that's the next subject. I'm sure now everybody wants to go to the refrigerator and get some food, so why don't we do that, and then we will resume. Okay.